I want to thank the Staglin family, and I serve on the Scientific Advisory Board, and this is my first year doing this, and I had the honor of reviewing the applications for the Rising Star Awards, and I have to say, it's really phenomenal to see the talent that we still have, that we're able to support, and this foundation is doing extremely important work by supporting rising stars. These are the individuals who are going to make a difference for what we're doing over the next 20, 30, even 40 years. So congratulations to the family and to the foundation for making a really wise choice in terms of all the things that they could have done. My charge today is not to talk about my own research. I'm going to talk about this idea that mental illness, at its core, is a disorder of development, and that just because the expression of the diseases that we are focused on, whether it's schizophrenia or bipolar disorder or adult onset depression, because they occur in the adult, doesn't mean that the roots are not present developmentally, and that the challenge for the field now is to understand how to put that in a context where we can utilize the information that we're generating from all the science that we're doing, both in terms of treatment now, as well as thinking about the future and how we're going to utilize this information to identify individuals who are at greatest risk and do things for them that are going to make a difference before the onset of disease. And you put this in some context. My oldest brother is a cardiologist, and if you talk to him about the way he's practicing medicine now compared to what he was doing 20, 30 years ago, is completely different. The way he's practicing it now is really what we're dreaming about, I think, in mental illness, to be able to identify components, signatures, perhaps, of individuals before the onset, in his case, of a heart attack, and deal with both the genetic risk, the lifestyle risk, and everything else that you can put together to create a path for individuals to follow that will prevent the disease. That's what's happening now in cardiology, and that's hopefully where we're going to be able to go here. So because I'm not talking about anything related to my own researches, which is neurodevelopmental disorders, and I focus on autism now, I am going to show you the newest arrival to the family. This is my poetic license. Now, the oddness is not that I'm a grandfather. Of course, that's odd. And Sam and I know each other for a very long time. I was a graduate student in San Diego when Sam was on the faculty there. The oddness is thinking about my son being a father. That's what's really remarkable. But it's not that I don't think about developmental issues on a daily basis when I see my granddaughter, Molly. They live in Los Angeles, so I see her quite often. And I have to catch myself thinking about what's going on in that brain. At this time, she's making about 700 nerve connections a second, 700 synapses a second. So there's a lot going on. So I want to start about this concept of neurodevelopmental origins of adult onset disorder in the kidney. This is a study that was published in 2003 in the New England Journal of Medicine, in which they looked at individuals who died, who had hypertension and died early. They died in their 40s. And they wanted to know whether there was just something fundamentally different about their kidney, which is sort of the origins of the problems with hypertension, the inability to filter all the fluid and everything going on in the body. And this is what the filtration unit looks like. You can see two of them up there that looks like little cauliflower buttons. And that's what it looks like in a typical individual. And when you look at somebody who died prematurely in their 40s and they had post-pubertal onset of hypertension, look at the cauliflower buttons now. Right? They're like twice as large. Right? It turns out in this study, not only are they twice as large, I'm going to show you data today. You're all going to be neuroscientists by the end of this 40 minutes, I promise you. That individuals with hypertension died prematurely in their 40s had half the number of filtration units, but each filtration unit was twice as large. So what's going on? Well, the kidney had no choice. It has half the number of filtration units. That's defined genetically. At birth, we have the total number of filtration units that we're going to have for the rest of our lives. That's defined genetically at birth, right? But those number of filtration units are going to have to handle the same load that all of us handle if we have twice as many. So somehow the system has adapted by making each filtration unit twice as large to be able to handle this fluid, but over time this load is too much to handle. It could be lifestyle. It could be all sorts of things that we don't know about that are hidden during this period of time when we don't know that this is at risk, and suddenly the system breaks down. This is, in essence, what we're thinking about in terms of the developmental origins of mental illness, that somehow the system is put together in a way that creates challenges over developmental periods through puberty, and that over time the system can't handle the load and breaks down. And I'm going to talk about what some of the evidence is for that, and we're looking in the future in terms of the kinds of studies that are being done in the field. So we know that there's a complex set of risk factors, both genetic and environmental. This is actually a diagram from Dan Geschwin's paper that was published focusing on autism, but it's a really nice illustration that we know now that there are all different kinds of genetic mutations, other sorts of changes to our DNA that are contributing to disease. But in bipolar disorder, in schizophrenia, we know that that's not the whole story. In fact, in all complex mental illnesses, we know that's not the whole story. There's a balance between that, between protective factors, and I put those in quotes because we actually don't know what those protective factors are, and the environment. I'll get to that in the next few slides. So think about genes as providing the blueprint for what I say vulnerability resistance by altering fundamental developmental outcomes. They kind of set the system up. In the case of the nervous system, the initial wiring is occurring before birth, but experience is used after birth to continue to drive that process. Sam will know this, that when I was a graduate student, I just felt that somehow I was investigating a miracle, right? Because the brain is different than any other organ system. The initial genes set us up to be able to gather information from the outside world and then continue to use that to continue to build the very organ that we're using to gather information from the outside world in the first place. That was pretty confusing, right? That was pretty confusing. So we have enough wiring together at birth to be able to take information in, and then we have to use that to continue to build the brain. The kidney, the liver. I love my kidney, both of them. Although at times I've said I'll donate a kidney for something that I wanted from my dean. My liver, my heart, I love all those, but the brain is very different, right? So the genetics get it wired up, but in the environment, the experiences that we have early on and through childhood and into puberty and beyond, that's part of the equation of what we have to figure out. So environmental factors participate in the process of tipping the developmental scales towards a better or a poorer outcome. And some of the policy work that I do, it's really about abuse and neglect. It's very different here, but the profoundness, the impact of early environment is really quite remarkable. And I'll show you why that is, because the nervous system, the brain, responds to that experience that it's having. And in some ways, you can think about it this way. It's thinking, well, this is what I'm going to be experiencing for the rest of my life. I better set my nervous system up to be able to deal with it. And sometimes that response, that change in the long run is not so good, right? And then in general, each individual, you think about gene-environment interactions, each of these, a genetic risk or an environmental risk alone is not enough to drive the system to end up with mental illness. It's the combination of the two. Genetic risk through mediating of atypical neurodevelopmental processes. There are 3 points that I want to make. Genetic risk through mediated of atypical neurodevelopmental processes. What does that mean? What that means is, as I already told you, there's a cast of characters, the genes that we're discovering, and some of the Staglin rising stars that you're going to be talking to during the course of the day, and I really encourage you to do that. For those who are speaking afterwards, as well as those who are sitting here, are making these discoveries, not like from year to year, but I mean from month to month. We're making discoveries that are creating that we know now are contributing to this landscape, whereas in fact schizophrenia, about 50%, if you look at studies of twins, both identical twins and non-identical twins and other genetic studies, about 50% of the disorder is heritable. Well, that's 50%. That means there's another 50% that's not heritable. Keep that in mind. But it's relatively high. Huntington's disease, for example, is 100%. So all risk genes participate in neurodevelopmental processes. That's the conundrum, right? So we have about 25,000 genes in our genome, and the brain is so complicated that it uses all of it at some point in time. The estimates are that about 80–90% of our genes are expressed in the brain at some period of time in development, right? So that means that any gene that's responsible for risk, or any combination of genes, you could assign to a developmental process, and we're trying to work through that now, but that's part of the challenge. It's not that certain genes are used just for one period of time in your life, but they're actually used over varying periods of time in your life and may actually perform different functions depending upon where you are in your lifespan. So I would say that the translation of the gene part, with all due respect to my genetics colleagues, is relatively easy. That is, we're identifying genes that we know increase risk for schizophrenia, bipolar disorder, depression, autism, you name it. We're identifying these genes, we can replicate it, and we're now getting to the point where we can identify even the kinds of mutations that are responsible. So it's relatively easy to get to the descriptions. And if I was giving this talk 6 or 7 years ago, I would say we're still struggling with even identifying who the players are, right? Who are they? But we're really made a lot of progress. So here we go. And you can't see this. I'm not going to go through this in detail, but the work of geneticists has revealed to us that it's not just common changes, a change that that all of us carry in a particular gene, a change in the sequence that changes how the gene might function or how much of the gene is made at a certain period of time. But it's also rare mutations that are private, that I may have, and I probably have many of them, that Francis may not have, but are nonetheless responsible in part for my pathology, at least according to my parents. So there's rare mutations, and there's what we now know as what's called copy number variations, which Lauren Weiss works on, who's here, in which there are deletions of genes or additions of genes which change the ability of the cell to function, or brain cells to function, and can have really profound and powerful effects on how the system gets wired up and how the system is able to function. So the second component, environmental correlates of increased risk, that's a scientific swamp. And I'm not exaggerating. I think it really is a scientific swamp right now, because your guess is probably better than ours as scientists, because we come with some baggage. We have our favorite environmental factors that we think are contributing to this. But I think, in fairness, we're so far ahead on the genetic side and so far behind on the other 50% on the environmental side. It's going to be a huge challenge to understand how you put these together. And we know we have to, because this brain, again, requires that interaction. So what do we know about? If you go back in the literature, there's descriptions of increased risk from non-genetic environmental factors that could increase risk for schizophrenia or other mental illnesses. So for example, first and trimester influenza or herpes simplex virus during pregnancy. But if you look really carefully, the effect sizes are pretty small. So does it increase risk? Yes, pretty small. Nutritional status during pregnancy, some very good studies during large populations during periods of famine in which there was an increase in the prevalence or the diagnosis of schizophrenia. And again, the effect sizes, they're there, you can see them, but they're relatively weak. So it's not that they're not unimportant, but they're weak. Obstetrical complications, it's not really clear. I mean, there's some studies that say yes, other studies that say we're not so sure. When you put them all together, relatively weak. I don't have good news about this part, by the way. When I said scientific swamp, I meant a scientific swamp. So this one is really intriguing, and Luann will like this, right? So the third area in which we're starting to make headway. So we've got genes, we've got a lot of gene candidates now. We've got environment, we know it's contributing, but we're not quite there yet in terms of our level of sophistication of how to investigate gene by environment. I'll talk a little bit about some advances that we've made in understanding how the environment can actually put a signature on our DNA material for a lifetime. which I think is very important to understand. Eric will be happy when I talk about that. But this issue about peri-adolescent biological environmental events that trigger disorder expression. So what does that mean? Well, scientists tend to be lumpers or splitters. We generally focus from a developmental perspective on very early aspects of development, prenatal development for the most part, in our model systems, or early periods during infancy and childhood. And then we skip in humans, about 20 years or 30 years, we go to adults. There's a lot of stuff that happens in between, and we know now from a lot of recent work that there are really dramatic changes that can occur in the wiring of the nervous system, even during puberty, during this period of puberty. Now, I say that to scientists, and they go, really? And then I say this to a parent, I go, well, of course. Right? If any of you are lucky enough to have a teenager who wakes up one day hating you and the next day sort of loving you, there's something going on in that brain. I know there's something going on in that brain. And so what do we know about these peri-adolescent periods? Because the brain is changing, and we think that this may be a contributing factor to the culmination of the developmental period in which we have genetic changes, environmental contributions that are affecting how the brain is getting wired up. Over time, think about the kidney again. The system is getting more and more challenged. Perinatally, during the peri-adolescent period, as the individual's physical characteristics are changing, that includes the brain as well. So what do we know? So we know there's reorganization of neural circuitry, and we know there's reorganization of neural circuitry in very important areas like the frontal lobe, the area that provides us with the ability to control ourselves, executive function, our ability to control our emotions, to put the cognitive and the emotional domains together. That's maturing during this period of time. In fact, some people accuse me of never having gone through that maturation state. There are neurochemical changes going on during this period of time. Right? So the brain starts developing prenatally, but it doesn't end at school age. It doesn't end at 5. It goes through this period of time, continuing to change, and even through puberty. And these reorganizational changes we think are important in contributing to this added risk and eventually expression of the illness. And we know also that the substance abuse and other teen stressors that are part of the mix actually are interacting with some of these changes in terms of reorganization of neural circuitry and neurochemical changes. So let me give you an example. This is work from, This is work from David Lewis's laboratory that was actually done about 12 years ago, and I think except for some of us who like to read the anatomical literature, we like to read the anatomy of the brain. David's group described some years ago that, in fact, the very system that we know is part and parcel of disrupted thought processes in psychosis, the dopamine system. That system in the frontal lobe goes through really remarkable changes in terms of its organization during puberty. It has expression patterns within part of the frontal lobe that look like this, and then over time, this part which you see in white, those are dopamine fibers, goes down, and there's an increase up here. Seems pretty subtle, but when you go in and look at it and sort of count, you see that there's some major changes going on in terms of where dopamine is having its influence during teenagehood and after. Well, that's not insignificant. That's an important change in that system that we know is responsible in part for the dysregulation of thought processes, and in fact is the target of medications, right? So this is a change that's happening we think is very, very important. Well, we know that in terms of adolescent stressors, that there's some pretty reasonable data now showing that the effect of cannabis use during peri-adolescence and its risk of psychosis actually has a larger effect size than some of the things that I talked about before in terms of obstetrical complications or prenatal risk. And if you look, this is a list from a review in which the effect sizes are anywhere between 1.5 to threefold increase in risk. Now, what's interesting about that, there it is there, I have my animation, I love my animation, is that the cannabis, the active ingredient, interacts with a neurochemical, a protein in the brain called the cannabinoid receptor, and that's present in the same part of the brain that the dopamine system is, and we think it's important in modulating both dopamine function as well as other neurotransmitter functions. And during the peri-adolescent period, this system is undergoing modification. And so exposure, if you think about this, if this is a hypothesis, exposure to this particular environmental stressor during this period of time when the brain is normally reorganizing itself, in the end, we think may, and we don't have any proof of this yet, but experiments are moving on this, may in fact change how the system is being redirected during this period of time. So you can see how there's an interaction of the environment with the normal process of development during the teen years that can drive this process in a direction that you do not want it to go. And there it is there. Okay, so what are the challenges and what are the opportunities? What developmental events are targeted and how do they differ across mental illnesses? So I've used developmental events in sort of a general term, but of course putting the brain together is a complicated process, and there are a lot of events that could be the target of both genetic and environmental insults. How do early developmental disturbances have long-lasting effects? I haven't told you how that happens. I've just told you that it does happen. Are there intermediate phenotypes that we can measure as predictors or precursor of disorder onset? Can we identify in individuals who are on this trajectory signatures that will help us to figure out where we want to put our money in terms of intervention and prevention? So what are the developmental events that are targeted and how do they differ across mental illnesses? Well, I would say the money is here. This is, I can test all the neuroscientists in the audience and say, okay, what is this? Eric, what is that? Synapse. It's a synapse. That's why he's chair of the Scientific Advisory Board. That's where all the communication takes place. That's the business end of things. I told you that during development, between the third trimester and about 3 or 4 years of age, we make about 700 a second. Snap your fingers, 700 a second. We also prune away a lot. Think about your garden. You let it overgrow a little bit. You prune it back. What's left, much stronger. And that process is part of how we use our environment and our experience to sort of drive that process. And it turns out that there's a lot of genetic information now that indicates that some of the genes that encode proteins that are very important for this communication process are risk factors for schizophrenia, bipolar disorder, depression, autism, you name it. Now, here's the challenge. The challenge is that depending upon who you read, that remember we have about 25,000 genes, about 2,500 to 9,000, depending upon who you read, at some point are expressed at that synapse. So that's anywhere between 10 and 40% of our genome, right, is used for building the structure and maintaining the structure and allowing the structure to function. That's used for communication. It makes sense in terms of how complex what we're trying to do. We're computing information in a very complex way all the time. So we're using a lot of our genome. So you don't need to be a statistical geneticist to figure this one out, that the odds of a mutation hitting a gene that has something to do with that biological function is pretty high. That's just the way it is. So that's going to be a challenge. But when you look at review papers in our field and you identify, there's lots of really interesting genes that have been identified. And so the one I want to talk about very briefly today is a gene that's been talked about before here, DISC1, disrupted in schizophrenia 1. It's not responsible for all of schizophrenia. There's some interesting structural changes in the gene that disrupt how it can function. What's interesting about this is that we've been able to move this from a candidate risk gene to demonstrating a biological process which is relevant to this neurodevelopmental origin thing. If we disrupt expression of this gene during development, what is it that occurs in the brain right, before puberty and after puberty that says, well, you know, this is a possibility. Disrupting these genes early can actually have these later onset changes in terms of how the individual, or in this case an animal model, will function. So I don't think about animals in our research as models for the disease. I think about them, I'm serious about this one, we're going to waste our time by trying to model the disorders that are uniquely human, but they're great for examining biological processes, specific biological process. So in this case, Niwa published a paper in a journal called Neuron, you won't be able to see this, demonstrated using a method that if you reduce expression of this gene, DISC1, in a mouse during prenatal development, just for a short period of time, right? It goes down for about 4 or 5 days and then it comes back up. That's enough to change that dopamine system that I talked about early on, right? It changes it early. You can see it before puberty, during puberty, and after puberty, you see the same changes. There's a reduction in the frontal lobe of a mouse, which doesn't have much of a frontal lobe, but it has something that we call frontal. There it is there, okay? You can actually see the changes here. I'll go up here. Very dark brown, very light brown. This is what happens if you knock DISC1 down, if you reduce its expression 3 months earlier, just for a short period of time. It's enough to change it forever. It changes expression of this neurotransmitter forever. That's not the most fascinating aspect. The most fascinating aspect of this study, I think, and again, it's not a model for schizophrenia. The most fascinating aspect, I think, is that they looked at certain behaviors that they were able to measure in the mice before and after puberty and demonstrated that even with these anatomical changes, the behavior that they measured, and it's something that we call pre-pulse inhibition. So, for example, if I were to do that, you guys jumped, right? I saw somebody jump. Actually, my back was turned. So that's a startle reflex. And we're able to inhibit that startle reflex if we, because we've learned how loud that sound is going to be. If we now experience a sound that's a little softer, and then hit that loud sound again, we're able to prevent the startle from occurring. And people with schizophrenia, it's not specific to schizophrenia or psychosis, have a difficult time doing that. That's a task. It allows the individual to kind of filter the sensory information coming in, and it's not done very well by individuals with schizophrenia. True with children with autism as well. They don't pre-pulse inhibit very well as well. Well, you can measure this in a mouse. You can measure this in a lot of animals. And they did that, and they said, well, before puberty, everything's normal. After puberty, not so good, right? That's good, that's not good, right? So everything's normal before puberty in that mouse that had this schizophrenia risk gene reduced. After puberty, it has this profound change. It's about a twofold change. So that's significant behaviorally. What's even more significant is that the use of clozapine in those animals, an antipsychotic, was able to reverse the abnormal behavior. Developmental onset change in a risk gene in terms of its expression, short period of time, right? Changes in the wiring of the brain, but nothing overt in terms of behavior. And then after puberty, we see these changes in behavior that we can measure, and we can actually reverse them with a relevant drug in terms of psychosis, in this case, clozapine. Now, there are other examples in the literature that I'm not going to talk about. What's the mechanism for that? Why is it that early developmental perturbation had this long-lasting effect? Well, we don't exactly know why, but I'm going to give you a hint as to why I think this is happening, and others in the field, I think, are thinking the same way. So how do they have these long-lasting effects? Well, I'm going to talk about something that's called the epigenome. Our own Eric Nestler is doing a lot of work in this now in its relation to addiction and depression, in which we know that we inherit DNA from our parents, but the DNA can get worked on even afterwards. It's not immutable. We can actually apply signatures to the DNA through experiences. And the experiences can be as diverse as malnutrition, emotional stressors, what we call oxidative stressors, immune challenges, all of these can have an effect and actually change the chemical nature of the DNA. And epi comes from the word above, so it's above the genome. These are changes that are occurring on our DNA, but are not occurring through an inherited process. Some of them are long-lasting. These chemical changes, they seem to be immutable once they happen. The memory, the signature, that's in pen. Others of these changes now we thought were immutable, but in fact can be changed. And I would say that's a signature that's written in pencil that can be erased. So the signature, what's important about the signature? Those chemical changes are important because they change when and where our genes are expressed. In the brain, that's very important. That's how we get ourselves wired together. When and where these genes are expressed, how we control that, critical for how the brain gets put together, critical for retaining the memory of what happened early on. So I have to show this because it took a long time to make. Let's see if Eric likes it. Okay, so there's a neuron. Okay, there's our brain. And that's the nucleus of that neuron. And there's our chromosome. And there's our chromosome unwound, and it's made up of DNA and other material, including proteins that are associated with the DNA, and control when and where the DNA as constructed as genes are expressed. So what happens? There's our external experience. That changes how the cell is going to respond to its environment, and new proteins are made through that experience. Those proteins actually then go into the nucleus and actually are part of the industrial complex to add on new chemical signatures onto the DNA, like putting a brake on the train that gets soldered or putting a brake on that can be released. What do you think, Eric? You like it? Okay. Okay. So how is that read out? Well, it's read out in terms of these developmental. So think of a stressor, and I'll show you an example of that in a minute. If you endure, at a very young age, high amounts of stress, that'll change the chemical signature of your DNA. I'll give you an example of that. Think back to this kidney, right? This kidney was granted half the number of filtration units that it should have had. And this is a diagram of any physiological system we have. We're always in this range of trying to maintain a stable state, whether it's our heart rate or respiration or whatever it is, we're always trying to maintain that state. When we're challenged over time, that's why we oscillate a little bit. We try to maintain that state within these boundaries, but sometimes those challenges, even acute challenges, can be very profound, and it changes the course of where our set point is, essentially. Our thermostat changes. And if it happens over time, particularly during development, we have a new set point. And this set point down here is completely different than what you saw up here. The new set point for the kidney, a glomerulus, that's a filtration unit that's twice as large, that, by the way, is a lot more difficult to maintain than a filtration unit that's half its size. So there's the load that we take on. We call it allostatic load. That's the load we take on developmentally, and it can change the course of how we're able to do business for a lifetime. So I'll give you an example of what we call these epigenetic changes and why it can have such profound effects. So these are experiments that were done by Michael Meany, Moshe Sif, Paul Plotsky when he was at Emory with Michael, in which they showed that differences in interactions between a mom and their baby pups can actually generate a change in how the animals respond to stress much later on in life. So these are what we call a high care mom. These are the high care pups. If you let them grow up and you look at their stress response, if we restrain them for a few minutes and we look at their cortisol levels, it goes up, it stays up for a little bit, it comes down. That's good. We want our stress response to come up, stay up for a little bit, and go down. We don't want our cortisol staying up there for a long time. It's not a good thing. When we look at other animals in which the moms provide much lower care in terms of interaction, well, I mean, they grow up, they weigh the same, they can do lots of things the same way, but when you look at their stress response, as adults, it goes up even higher, it stays on for a longer period of time, it takes a while to come down, right? Think about that one event and think about that over and over and over and over and over again, right? So what the group was able to show is that for a particular gene, which is known as a glucocorticoid receptor, which is responsible for shutting that stress response down, another flag is added here, that red flag. That's the chemical signature that goes onto that receptor, and it stays there forever. So these changes occur just after birth in the lifetime of the animal, right? But we see the effect long out into the animal being an adult through death. And we know now that this actually occurs in humans. It was first described in animals, and recent work shows that, in fact, this happens in humans as well. I'm not going to go through that data. So the developmental roots of health disparities, how adversity is built into the body, in part it's going to be through these processes in which we're changing the chemical signatures of our DNA. We're discovering almost on a weekly basis how this is controlled, which means that we're getting closer to the point where we can understand how the genetic disparities through inheritance, through what we inherit from mom or dad, combined with changing the signatures of our DNA over time through experiences, are affecting how the brain is being put together and wired, so that ultimately we're going to get closer to understanding the mechanism through which this happens and why it is that developmental problems can lead to adult onset disorders. So think about early adversity, whether it's genetic risk, environmental exposures, or malnutrition. This gets embedded in the biology of the individual early on. It doesn't mean it's immutable. It just means that it's getting embedded. Can it be changed? Absolutely. There's a lot of evidence for change. Even in the adult, there's lots of evidence for change, but it's happening. There's a biological change that's going on through these experiences. It's not without cost, though. There's a cumulative burden that these systems have to carry. Think about the kidney, right? It's okay for a while. It's dealing with this burden over a long period of time, having to filter all this fluid with a system that's not set up for filtering all of that fluid. So it's what we call allostatic load, physiological disruptions, neurodevelopmental, immune, metabolic, neuroendocrine. By the way, these systems are not separated in silos. They actually interact with each other, right? So what I'm talking about in the context of the developing brain is happening to other systems as well, including our immune system and how our immune system responds as adults. And then finally, what does that lead to? Health-threatening behavior, mental illness, physical illness, and to me, one thing that often is not discussed, but is clear from the literature, right? Mental illness leads to shortened lifespan, right? Probably through the cumulative burden over time, all the other components of an individual, their physical health, their mental health, all combining to create even greater risks that we really have to be dealing with. So we're focusing more and more, I think people are focusing more and more on how these systems interact and how the developmental burdens end up leading to systems that are at risk, both in terms of mental illness risk and physical illness risk. So some final thoughts: experimental challenges and opportunities for the translational neuroscientist. That's us. Okay, so what are some of the things that are happening now that we think are going to lead very rapidly to gathering information that's going to allow us to move from hypothesis to mechanism to translating that into clinical treatments and new ways of thinking about, that is 21st century ways of thinking about how we're going to deal with very difficult problem for the individual with mental illness and their family as well. So in experimental terms, we're getting much better at thinking about how and what we're going to measure and what we call an ethologically appropriate way. There's certain things that we do that rats don't do. For example, if you're interested in the vomit reflex, don't use a rat. They don't vomit. Now, you may be interested in figuring out why it is they don't vomit and trying to apply that to humans, but there's an example of something that you would not investigate in a rat. So we're getting much better at identifying those behaviors, including what we call intermediate, what I call an intermediate phenotype, following something early on, longitudinally. You'll see that's one of my triangle points, to get to where we really understand how it changes over time. Genetic modifiers. So we're using it now, in a sense, to identify risk factors. But we're moving much closer to the point where we can begin to look at genetics in a way to understand how it's contributing to the difference in treatment response, and other factors that we know are going to be very important for individuals with mental illnesses. And we're getting close to the point where we're going to be able to put this together because genetics is moving so rapidly. We can sequence your entire genome now in about 4 days, 4 or 5 days, and it's not so expensive anymore. So in terms of thinking about as risk, it's one thing, but thinking about it in terms of how it's affecting treatment response. I respond badly. Francis responds well to the same treatment. We have the same diagnosis. Why is that? And we need to move closer to that. Development is a longitudinal continuum for study. Well, that's a complicated way of saying cross-sectional studies are just not getting us where we need to be. And more science is being done where we follow you for a long period of time. And that's really important because we can see how you're changing and how you're responding, or how you might respond to certain treatments. Regulation and patterning of gene expression. I had to put that in. I can't emphasize this enough. In the past, we were trying to interpret genetic risk based on no information about when and where these genes are expressed that are wiring the brain together. And 2 years from now, we're going to have a database that's going to tell us where and when genes are expressed in the human brain from fetal ages all the way up through adulthood. That's an enormous advance in terms of how we're going to be able to use it. And it's going to be free access and open to every scientist around the world. Examination of developing circuitry and their individual components. Environmental impact, sensitivity, specificity, and time. We have to get out of that marsh. And how we're going to do that maybe is a focus of conversation today, because I don't have great answers to that. Thinking about these systems interacting in these disorders, neural and non-neural system interactions, puberty and the changing neurobiology. I can't emphasize that enough. We're moving towards that. People are beginning to understand this is a period of time that we have to examine, because this is going to give us clues in terms of who's going to be most vulnerable to disease onset and who's going to be able to withstand the challenges during that period of time as the brain is changing and as the environment's changing. And I want to bring this up. This is my last slide. Tom Insel, who's the director of NIMH, 3 years ago when I was on council, the National Advisory Council for Mental Health, asked myself and John Marsh, who's a pediatrics, who's a child psychiatrist at Duke, to chair a committee to say, okay, if we believe schizophrenia and bipolar disorder, adult onset depression are developmentally based, how much are we investing in this as a country in terms of research? And the answer was, sadly, very, very little. So we convened a group of people and we wrote a paper, which you can download for free. This is a report of our work group, which developing a field called transformative neurodevelopmental research, or translational developmental neuroscience. Clinicians, scientists working together. This paper has ended up resulting in something like $150 million new dollars from the NIMH, specifically targeted to try and understand the neurodevelopmental basis of mental illnesses. And it's an example of scientists getting together, understanding that there's core components of what we're trying to solve that are not being investigated, and then changing the course very rapidly in terms of where we're putting our money. And there's a lot of new projects that have come up in the last 2 or 3 years that are being funded through this initiative now, and it's going to continue not just in mental health, but in other areas as well. Thank you. I think I'm on time, maybe a little late, but thank you very much.